Join. So, hello everyone. Good afternoon or morning. It's a pleasure to introduce Ekaterina or Katya Osipova, currently a doctoral researcher at the Harvard University. She's an evolutionary biologist working at the interface of genomics, behavior, and adaptation, with a strong focus on how extreme phenotypes arise in nature. In the past, her research explored a wide range of fascinating systems, from metabolic adaptation in nectar-feeding birds to the remarkable physiology of hummingbirds. Always using comparative genomics to understand how evolution repeatedly finds solutions to similar ecological challenges. More recently, Katya has become the lead researcher on a project funded by the National Science Foundation, which investigates the genomic basis of brood parasitism in birds. Her project aims to uncover the genetic and regulatory changes that underlie this transition in life history. Before giving her the floor, I would also like to mention that this is our final special interest group talk of the year, and we are already planning the 2026 speaker lineup. So if anyone is interested in presenting or wants to nominate a colleague, self-nomination are very welcome, and you just need to reach out to us during this talk on the Slack channel of the animal-genomics, or just send us an email. So we are very happy to have Katya with us today. Welcome, and please take an imaginary seat in our stage.
All right. Thank you so much. That was a very comprehensive introduction. I did not expect that. Thank you. And thank you again for giving me the chance to talk about my research. This is the work that I've been doing for the past several years. Some of it is like a little bit from probably like a couple of years ago, and some of it is very, very recent. So basically the experiments were done yesterday. So yeah, I'm very excited about that. And as Francesca mentioned, I had a chance to work on a number of projects in different areas, but metabolism has been the central driver of my research interests. And all organisms on Earth face a fundamental challenge of how to extract energy from the environment and convert it into forms usable for life. The sum of the processes of addressing this challenge is called metabolism, and this is a fundamental and a universal property of all living systems, and yet most of the metabolic research has been done in just a few model organisms, like humans and mice. And it gives us a very limited perspective of what metabolism is capable of and what are the true drivers of metabolism. In the end, it poses a huge barrier to understanding of our own metabolic challenges, as well as developing tools to manipulate metabolism for any sort of biomedical applications. At the same time, for nearly every human metabolic challenge, there would be an animal that naturally solved it. Like, for example, hummingbirds, they consume immense amounts of sugar and they don't suffer from diabetes. Ground squirrels, they hibernate, stay immobile for months, and never lose muscle mass. Seals and whales are technically obese, and yet they're still perfectly healthy. So animals demonstrate a huge diversity of metabolic adaptations. And when we focus on just a few model organisms, we are covering only a couple of data points in this vast metabolic space. And in the end, the rest of metabolic diversity remains unexplored. The overarching goal of my research is try to explore this metabolic diversity in animals and see how we can use this knowledge to design potentially novel metabolic systems that are pushing the limits of the natural systems. And to get there, I'm addressing three fundamental questions. First, what is the genomic basis of metabolic adaptations? Or if we want to put it simpler, if we're starting from human metabolism, how we can genetically jump to, let's say, hummingbird metabolism. Second, at which level of biological organization we can see the differences in metabolism. Is it organismal, tissues, cells, or subcellular level? And finally, can we use the knowledge of the natural systems to try to jump into undiscovered niches yet, so push the limits of the natural systems? And in the past, I was focusing on the first two questions. This is what I will be covering mostly today in my presentation. And by the end only, I will try to give a little bit of an outlook of how we could address the third question. So I structured my presentation in a way I will be talking about two main stories. In the first one, that is more specific, I will focus on the genomic basis of extreme metabolic performance. But next, I will try to put it in the broader context and try to address the question of convergence of extreme metabolism. When we're talking about the extreme metabolism and pushing the limits of natural systems, as I mentioned, hummingbirds, they are the first that are coming to mind. And to give you some impression of how insane their metabolism is, they can consume up to 3 times of their body mass of flower nectar a day. In human equivalent, that would be around, I don't know, 200-liter bottle of Coke. They process the sugar really fast, and they manage to directly fuel their flight from the newly ingested sugars. They are the only birds who evolved through hovering flight. And to demonstrate you how special this type of locomotion is, I have this little video. So as you can see, hummingbirds are extremely fast. They can flap their wings 88 times per second. And this is a slowdown that shows how exceptionally maneuverable hummingbirds can be. They can hang in place, they can go forwards, backwards, up and down. And while hovering, they demonstrate the highest metabolic rate among vertebrates. And in this project, we were trying to answer the question of how hummingbirds can have this metabolism from the genomic viewpoint. And we opted for comparative genomics approaches, trying to be more systematic and not to focus on any specific candidates. And to make use of those approaches, of course, we need genomes. When we're thinking about metabolism of hummingbirds, we notice that both nectar feeding and hovering flight, they are ancestral traits of all hummingbirds. They are present in all hummingbirds. So likely we're looking for changes that happened in the ancestral branch of all hummingbirds. So we started with generating a new genome of a long-tailed hermit. A little technical plot. This is how the assembly of the long-tailed hermit looked like. In brief, the higher the line is, the better from the contiguity viewpoint. And as you can see, the long-tailed hermit exceeds the contiguity of the other hummingbird assemblies and also the reference chicken assembly. So together with other hummingbird assemblies, we managed to span the entire hummingbird clade and to accurately identify changes that happened in the ancestral branch. And of course, we're looking for changes that happened in hummingbirds, but not in other birds. And to have something for comparison, we in total had 50 bird genomes. And we managed to run a bunch of comparative screens. The one that we found something interesting in was the screen for lost genes, when we were searching for inactivating mutations and identifying the genes that were pseudogenized in hummingbirds. We found a bunch of them, but only a few were shared between all hummingbird assemblies that we considered. And that means those are the candidates that are lost in potentially in the ancestor of hummingbirds. And 4 of those 5 genes, to my knowledge, don't have to do anything with metabolism. But the 5th one, FBP2, encodes fructose 1,6-bisphosphatase, and I will go into details what the gene actually does. But for now, this is a gene that is crucial for sugar metabolism. So we're wondering, maybe this is something that is relevant for hummingbird metabolism. Before jumping into gene function, what we saw, what happened with FBP2 in the birds. So FBP2 is represented by 7 coding exons. And in hummingbirds, we saw that this gene is hammered with inactivating mutations. Also, those highlights basically mean the inactivating mutation that happened in this exon of the gene. Like, for example, the stop codons, with a lot of those mutations shared between different hummingbird assemblies, like, for example, the stop codon in the first exon, several splice site mutations, and the frame shifting insertion. This suggests that FBP2 was inactivated in the ancestor of all hummingbirds. We also confirmed that using molecular dating approach, and we dated the loss of FBP2 to happen around 40 million years ago. The interesting thing, we can also combine it with the data on the fossils that are showing when hovering flight evolved. So the fossils are roughly outlining how the bird looked and therefore how the flight of the bird could look like. And we have an estimate that the FBP2 loss potentially overlap the evolution of hovering flight. In fact, there are 2 FBPases that perform similar functions in a tissue-specific manner. At least this is something that we know in mammals, with the paralog of FBP2, FBP1 performing its function in the liver, and FBP2 performing its main function in the skeletal muscle and heart. We generated transcriptomic data, and we wanted to see if this is something that is also true for our birds. What we see in the non-sugar-feeding close relatives of hummingbird swifts is a nice recapitulation of the mammalian data. So FBP1 is highly expressed in the liver, and FBP2 is expressed in pectoralis, or the flight muscle. When we looked at hummingbirds, first of all, we noticed that FBP2 is not expressed anywhere. That's a good sign. An activation, no expression. But the other important thing is FBP1, a paralogous gene, is not expressed in the flight muscle either, meaning it doesn't take over the function of FBP2, and there are no functional FBPases in hummingbirds. So now we know that FBP2 is inactivated in all hummingbirds, not expressed, what that could mean for them. To get there, I want to take a step back and see what gene loss can actually contribute to evolution. And gene loss can be lost according to different scenarios. One is the most common, is we're not using the gene, so it's being inactivated. Like, for example, this is something that happened in subterranean mammals. They went underground, they didn't need their eyes, and a lot of genes involved in eye development were just lost. The second scenario, when the gene loss can actually promote adaptive evolution, is very rare, but it happens, and there are a couple of examples in the literature, like, for example, the inhibitor of lipase that was lost independently in different lineages in herbivorous groups could promote fat digestion in those mammals. Or the loss of the inhibitor of insulin secretion in fruit bats that could enhance insulin secretion in species that are feeding on a lot of fruit. So the question is, which scenario it is for FBP2? And for that, let's first take a look at the function of the gene. In a cell, glycolysis and glyconeogenesis are 2 tightly interconnected processes. Most of the reactions are being shared between those 2 pathways, with the exception of the few that are highly controlled and usually rate limiting. And FBP2 encodes the rate limiting enzyme of glyconeogenesis. So if hummingbirds lost FBP2, potentially it could lead to very tangible consequences for their cellular sugar metabolism. And to test that, we actually went and tried to do the experiments, do the functional validation of how it works in cell systems. So I used the bird myoblast as the model system and used the CRISPR-Cas9 approach to target and delete the functionally critical exon 5 of the FBP2 gene with the combination of guide RNAs. Now, again, a little bit of technicalities to make sure that we are actually doing what we want to do. I managed to produce a pool of cells with the FBP2 that is knocked down. And as you can see, the wild type is just the expected band for this region. And with the FBP2 knockdown pool, we see a number of bands that are corresponding to expected bands with the combination of guides for this region. And of course, I also validated that FBP2 is downregulated in our cells. And sometimes it is possible that there is some kind of the genetic compensation for the gene that is being inactivated, and the paralogs of this gene are trying to take over the function. And what I also confirmed is FBP1 is not upregulated in the pool of the knockdown cells. So now we have a system of myoblasts with the knockdown FBP2. Let's see what happened to metabolism of those cells. And there are different ways to assess the cell metabolism. One of the most common, the state-of-the-art method, is seahorse. It's kind of a complex scheme, but I will walk you through what the experiment actually shows. Energy is being produced by two main pathways in the cell, glycolysis and mitochondrial respiration. And by adding specific inhibitors for both glycolysis and mitochondrial respiration, we can assess the contribution of each pathway by the external signals like consumption of oxygen and extrusion of protons. So to give an overview of how the experiment looks like, we have our cells, we feed them, and then we measure how fast they consume oxygen and how fast they acidify the media or extrude the protons. So what we saw in our cells, first of all, the downregulation of FBP2 in cells increases glycolysis, increases the glycolytic capacity. The next thing, in the same set of experiment, we also saw that it increases mitochondrial respiration, so the oxygen is being consumed faster in those cells. With the glycolysis, it's kind of obvious what the connection to glyconeogenesis is, but with mitochondrial respiration, like, why does that happen? Can this be related to some downstream effects, like, for example, the increased mitochondrial number? And indeed, it does have to do with that. So we can measure the mitochondrial content in the cells by the mitochondrial to nuclear DNA ratio. And as you can see, the downregulation of FBP2 actually increases the number of mitochondria. With our transcriptomic data, we also can see if this is something that reflects the real organismal level data. And comparing the expression data in swifts and hummingbirds, we see that hummingbird muscles have higher mitochondrial gene expression. And this is not the case for the hummingbird liver. So that means that not only glycolysis is being affected by downregulation of FBP2, but with downstream effects, it also increases the number of mitochondria. To summarize everything that I talked about in this part of the talk, all hummingbirds lost a key glyconeogenic enzyme, FBP2, and it promoted higher glycolysis and higher mitochondrial respiration. To bring up the analogy in a way of like, we are releasing the brakes in the car and we're allowing the car to run faster. It is also interesting that in many cancerous cells, people also report downregulation of FBPases with very similar effects of the upregulation of glycolysis and contribution to the effect that is well known in cancerous biology, Warburg effect. But hummingbirds are not the only ones that evolved the adaptations to high sugar feeding. They are definitely among the most extreme because 90% of their diet consists on flower nectar. Those are the birds that are spread in North and South America, but there are other bird groups that are less well known, like, for example, sunbirds that are inhabitants of the equatorial regions of Africa and Eurasia, and also lorikeets and honeyeaters that are endemics of Australia and New Zealand. And all those birds, they rely on flower nectar a lot in their diet, and they share the efficiency with what they process the ingested sugar. And this brings me to the second part of my talk, the larger question of convergence of extreme metabolism. A lot of birds, they occasionally take something sweet in their diet, either fruit or nectar. But those four groups that I talked about, they stand out and they spread across the phylogeny like you can see here, suggesting that they independently evolved adaptations to nectar feeding. So this prompts the question of what is the genomic basis of this adaptation? And second, and probably even more interesting, if the genomic solutions that these birds are going for are repeated or convergent, or they are lineage specific. And this is a highly collaborative effort. This is the project that we were running with our core team with Maud, Maggie, Michael, and Tim. And I want to say a huge thank you. I think we managed to produce work we can be proud of. So when we're talking about genomic changes underlying phenotypic variation, I mentioned one, the gene loss, but obviously this is not the only one that we can be looking for. And in this case, we were trying to be as unbiased and systematic as possible. And we were looking for signatures of any selection. And we were looking for changes that happened in the genes, as well as the changes that potentially happened in the regulation. In this case, those could be the changes that happened in the regulatory regions, or just the changes in the expression of those genes. So again, to be able to make use of the comparative genomics approaches, we need genomes. And in this case, not a lot of genomes can be found outside of the hummingbird clade. So we started the project with generating a bunch of new genomes for nectar feeders and their non-nectar feeding outgroups. We generated 9 new genomes that are highly contiguous. This is a simplified plot, but the bar basically means how contiguous the genome is. And probably everyone on Zoom knows, if we're sequencing the genome, we're ending up with like a huge string of ATGCs. And from the functional viewpoint, it doesn't tell us much. So the next step, the annotation, is absolutely crucial. And now it's still a bottleneck in the comparative genomics analysis. So if we're trying to get the publicly available data, we'll find this for only half of the species that we're considering. And to address this bottleneck, we developed TOGA. This is a machine learning-based method that uses the annotation of a reference genome, the one that is well annotated, and based on the genome alignments, tries to project that annotation onto other genomes. So to be even more comprehensive, I combined the toggle predictions with the other sources of data, including transcriptomic data, protein data, and even de novo predictions. In the end, I annotated 19 genomes for this project, and from the completeness viewpoint, they are all highly complete, nearing 95 and above percent of completeness. Finally, to be able to see what happened with the gene expression evolution related to nectar feeding, we also sequenced a number of transcriptomes of the nectar feeding species together with their non-nectar feeding controls for 5 metabolically relevant tissues. So in the end, that's a lot of data, and we managed to run a number of genomic screens. And today I will be focusing just on a few highlights. But first, I want to give you an overview of the entire analysis. Across the genes, we screened 11,000 one-to-one autologs for signatures of positive selection. We screened 300,000 CNEs, or conserved non-coding elements, as proxies for regulatory regions, and also the same set of orthologous genes for each transcriptome that we acquired to see the expression changes. Final thought before I jump in the results. When we're talking about convergence, there are multiple levels we can detect convergence in. And first, the most obvious, when we see the signal of selection in the same gene in different bird lineages. And this is something that we refer to as molecular convergence. But it's also possible that in the same pathway, different genes are affected, like as you can see here. And in this case, we're talking about a higher level of convergence, or pathway level convergence. And this is something I want to start with. So at the pathway level, what we see in our nectar feeding birds is convergent signal of selection in a few pathways that are potentially very interesting for their metabolism. Some of them, they are expected. So like, for example, pathways or terms that are associated with energy homeostasis or carbohydrate metabolism. But some of them, they caught our eye because they were not immediately obvious. Like, for example, those two, regulation of system process and cardiac muscle development, they include in genes that are involved in heart performance, heart development, and blood pressure regulation. And interestingly, this is also the pattern that we see in regulation in CNEs. Again, we see the pathway that is affected, that is involved in regulation of blood pressure. The question is, why does that happen? And that was a little bit puzzling at first, until we actually see what the effect of the glucose and fructose on blood pressure is. This is something that is known for mammals mostly, but both fructose and glucose, they upregulate, they increase vasoconstriction and increase blood pressure. They work through different mechanisms. Glucose work through the insulin pathway, and fructose likely through other mediators like uric acid and nitric oxide. But the result is the same. The excessive sugar consumption leads to increased blood pressure. And potentially this is the challenge that all nectar feeders have to solve, and this is how they address the challenge convergently. Now, at the gene level, at the level of molecular convergence, we actually didn't see much. So we detected exactly one gene that was under positive selection in all nectar feeding lineages. But it was a very interesting gene. It's called MLX-IPL, and it is involved in both sugar and lipid metabolism. To be more specific, it encodes a transcription factor, which performs its main function in intestines and in the liver. It's being activated by metabolites of the glucose, where it triggers its translocation into nucleus. It pairs up with its interaction partner MLX, and together they are triggering the driving the expression of glycolytic and lipogenic genes. So this is something that is potentially very relevant for the metabolism of birds that are feeding on sugar. With our expression data, what we see with MLX-IPL is this consistent, very significant upregulation in both tissues across all nectar feeders. So MLX-IPL is definitely affected on the sequence level, protein sequence level, and at the regulation level. Considering that this is kind of the upstream point of a lot of changes, we were also wondering what happened with the downstream targets of MLX-IPL. So from the expression viewpoint, the interaction partner of MLX-IPL is also upregulated. But the most interesting thing, all the genes that's supposed to be triggered by MLX-IPL, they are also upregulated in all nectar feeders. Especially this is obvious with lipid metabolism, with the crazy upregulation of lipogenic genes in hummingbirds. Is this something that is also reflected on the higher levels, like on the tissue level? And there is a way to look at the lipid deposition in tissues with the special dye. It's called oil red O. We managed to obtain tissue sections for hummingbirds and for non-nectar feeding zebra finches. And first, in zebra finches, what we see in their livers is something that is expected, like, for example, for humans. So those little droplets of red that you see here, those are depositions of lipid in the liver. And I will give you just 1 second to think about what we saw in hummingbirds. But I think at this point, when I talked about the upregulation of the lipogenic pathway, it's pretty obvious. So yeah, hummingbirds have extremely fatty livers. All those red droplets that you see, this is like the entire tissue covered with fat. And this is something that is pathological for humans, but apparently adaptive in hummingbirds. So in our birds on different levels, we see those changes that are triggered by MLX-IPL. We were wondering if we can go down and actually have a look at the changes that are contributing to those effects. Can we see something? And if we see at the alignment, the protein alignment of MLX-IPL, we see a bunch of changes that are driving the signal of positive selection in nectar feeding species. And interestingly, a lot of them, they are falling into the regions that are called glucose sensing domains. So potentially this is something that changes the function of MLX-IPL on the molecular level. To test that, we again went into functional validation, tried to do the experiments. In this case, we synthesized two proteins, MLX-IPL together with its interaction partner, for two birds, hummingbird and the swift. And we introduced those constructs in human cells, where we could test how well the proteins coming from birds are performing the function that MLX-IPL is supposed to perform. So how well it binds to corresponding elements and how well it drives the expression of the downstream genes. And this is the work that was done by Konstantin and Mikhail, our collaborators at Charité. So huge thanks to them. First of all, with the binding and the activity of MLX-IPL, we see that the hummingbird version of the protein is slightly better than the swift version of the protein. But this difference is significant to drive a significantly higher expression of the MLX-IPL target genes with the hummingbird protein compared to the swift protein. So what we see here is the hummingbird version of MLX-IPL enhances sugar response in cells. Now to summarize everything that I talked about in this part, we first of all found the positive selection in genes associated with heart development and blood pressure regulation. And I hope I convinced you how this could be relevant for birds that are feeding on a lot of sugar. Second, we found that MLX-IPL, a transcription factor, is under positive selection in all nectar feeders. It also has this convergent upregulation and expression. So potentially this is this very cool convergent point, the knob that every nectar feeding bird is supposed to tweak to actually feed on sugar. With our functional experiments, we showed that MLX-IPL of hummingbirds drives higher sugar response in cells. And we saw a huge downstream effects with our transcriptomic data on the lipogenic genes with high expression of lipogenic genes in the liver that is also reflected on the phenotypic level in this very extreme way with insanely fatty livers of hummingbirds. And finally, I had a chance to talk about just a few highlights, but in general, there were a lot of other screens, a lot of other results that we managed to see with this data. And in general, it's 9 new genomes, 19 new annotations, and 19 new transcriptomes that could be extremely valuable for any further studies of evolution and adaptation. So finally, I want to come back to my original questions. Today, I had a chance to talk about the genomic basis of extreme adaptation to excessive sugar consumption and the different levels where we can detect this adaptation. What I didn't have a chance to talk to is how can we use this knowledge to try to develop something new. But as you can see with the examples, with the experiments, with the knockout of FBP2 or introduction of the bird version of MLX-APL, it is very possible to introduce something that is coming from birds and actually get something that is gain of function in other systems, including human systems. Extreme sugar feeding, this is just one of the adaptations that we can study like that. But there are plenty more of metabolic adaptations that could potentially be transferred into some actionable items for designing novel metabolic systems. And this is something that I'm very interested. Currently, this is the research program that I'm trying to develop. So stay tuned. I'm very excited to probably tell you more results in a couple of years. So just before I finish, I want to say a huge thank you to all the people that helped us with this project, especially the core team of our collaborators. We managed to meet for like every other week for probably 5 years to manage to finish this project, but also a lot of other people that contributed to sample collection, sequencing and assembly, or histological work and the experiments. Yeah, huge collaborative effort. I think very cool work. Thank to all the people that contributed to this work, and thank you for listening. I am happy to take questions.
Okay, so thank you so much, Katya. I do have a question, but it's like just a curiosity. And I wanted to ask you what first drew you to hummingbirds and nectivores, birds in general?
Could you repeat the question? I feel like I missed the beginning.
Oh, okay, I'm sorry. Maybe it's my Wi-Fi connection, but I wanted to ask you what drew you to hummingbirds and nectivores?
Yeah, so it started with my PhD, and honestly, I was always obsessed with the metabolic pathways, how insanely complex, but at the same time efficient and logical they are. And I learned about, oh, actually, there are birds that have an insane metabolism. And apparently, at this point, no one actually tried to find out what happens with their metabolic pathways. Mostly because the genomic data did not exist. When I was starting my PhD, it was like one hummingbird genome, and we actually contributed to refining that hummingbird genome. And yeah, let alone I'm not talking about like the genomes of other birds from other clades like honeyeaters, sunbirds, and parrots. So it's the lack of the genomic data that didn't allow people to answer those questions before. But there was a lot of work on physiology of hummingbirds. A lot of people were trying to measure their respiration, try to uncover how they manage to hover that efficiently, like the mechanics of their flight. So it's the combination of like, we have a lot of the phenotypic data and we see how insane that is. And we know barely anything from the molecular viewpoint. And I thought like this gap is a very exciting gap that I could bridge. So I guess this is how it started with those projects.
Yeah, that's super interesting. So let's see if we have some questions from the audience.
If there are not, I have one. I don't. Okay. And there is some.
Okay.
I have a couple of curiosity. First of all, thank you very much, Ekaterina, because you explained very clear a topic that is not mine. I am a student from Italy and related to different things. And my first curiosity was about, because at some point you showed a fruit-eating bat that is a mammal. And I was wondering if you know or if you think something about the pathway of eat fruiting or sugar feeding mammals instead of birds.
Yeah.
And the second one, because you were very clear, but there is something still unclear for me, and it is, what do you mean when you try to respond your question about new niches?
Right. Yeah. Yeah, very interesting questions. Let me try to answer the second question first. So when we look at the metabolic diversity in animals, we are covering a lot. Obviously, it's a lot more than we can see in, for example, in model organisms like humans and mice, right? But my argument is this is not necessarily everything that is possible. And second, it's not necessarily even the best that it's possible. So if we are talking about like the fastest metabolic rate, we are talking about like very fast hummingbirds. Does it mean that it's actually the fastest animal that could exist? I would argue no. So this is what I'm talking about when I'm saying like undiscovered niches or novel metabolic systems. So in a sense of like, we see a lot, we know what's responsible genetically for the diversity of that trait or that trait. And potentially we can also use this knowledge to design something that goes beyond those limits. If this is metabolic rate, then we can try to generate systems that have higher or lower metabolic rate compared to everything that exists in nature. Yeah, I'm talking specifically about metabolic rate because this is something that is very quantitative, and it's easy to say, like, let's do something that is faster than anything existing in nature. But it's not necessarily the only trait, and you can imagine other things that people can study. I don't want to throw in like buzzwords that are like really actually difficult to test on the cellular level, that I know a lot of people are trying to address the question of longevity, for example. And some people are even stating like, this is something that we can see at the cellular level as well. Questionable? I don't know, maybe. But potentially first of all, knowing what's responsible for the genomics of the trait from the genomic viewpoint for the phenotype could help us to try to go beyond of what exists in nature. So this is to your second question. With the first question, could you remind me what it was?
Yes, it was about the bats that you showed before.
Yeah. Yeah, I know that now, like by now, there are a couple of new studies that's been released on the adaptations in sugar-feeding bats, so nectar-feeding bats, fruit-eating bats, and the pathways, including how they adapted their insulin response. So one of the reasons why I didn't talk about them that much is we decided to focus on birds because mammals and birds initially have already very different ways to metabolize sugar. And when I'm talking about like differences, there is a conceptual difference in the insulin signaling. Mammals are insulin responsive. So we eat a meal and then insulin rises, and when we can drop the blood sugar levels by incorporating the sugar in the cells. This is not something that's happening in birds the same way it happens in mammals. Birds are mostly insulin resistant, so they are just like slowly incorporating sugar from their blood. So from like the very basics of how sugar metabolism happens in those two groups, there is already a difference. And we decided to focus on birds just not to miss the specific things that are happening in birds, but not in mammals. So like the changes that would be specific for birds. But it doesn't mean what we find in birds is not relevant for mammals. Like, for example, MLX-IPL. It's the transcription factor that is studied well in mammals, and it does basically the same thing. It like regulates the lipid and sugar metabolism the same way it does in birds. More specifically about your question, I do know a little bit about how bats do that, but my focus is on birds, and those are the reasons why we decided to focus on birds, if that makes sense.
Wow, that's fascinating. I thank you very much.
Thank you,
Ekaterina.
Okay, so if there are, yeah, Francesca, I don't know, probably is having problems with the sound. If there are any other questions from the audience, if not, I have one. So my question is, so you mentioned that the next step would be to design ways to translate these findings into humans, but do not these kind of changes in the original species have compensatory mutations for some deleterious effects that they could have, like for instance, this high lipid reservoir in the liver that you were mentioning and stuff like this? So how will you control for this?
Yeah, absolutely. I mean, that is a great question, and this is the one that I thought about a lot. So basically, when we're changing something, how can we be sure that it's going to be incorporated in the existing environment, in the existing pathways efficiently? First of all, with the example of MLX-IPL, we can see it is absolutely possible in a way of like, the only thing that we have to think about is the immediate interaction partners. Like, for example, if it's a protein that is working in a complex with other proteins, then of course you'll have to think about like what compensatory mutation is supposed to happen in the other subunits in the other proteins that are interacting with this protein. But very often that's the only thing. So it's not necessarily you have to change everything in the cell for a specific protein that you introduce into function the way that you expect. But this is absolutely something that we can only answer by doing the experiments. So I would never say like, okay, now we're introducing this new version of this protein, and this is how metabolism will change. All right, that was not a question, I guess.
Yeah, someone mute himself.
Yeah, hopefully I managed to answer your question. So we absolutely have to consider that. By doing the experiments, by predicting what interactions this specific protein, regulatory element, etc., would have, But it is possible simply because we usually don't have to change everything in the cell. We just have to think about the immediate connections.
Okay, thank you very much. And I have another question that maybe I didn't get it. What is the role of this lipid reservoir in the liver in the nectar eating birds?
Yeah. So my assumption is this is the way hummingbirds store energy. There are multiple ways to do that, and one is storing energy in form of glycogen, the other one in form of lipids. And if we compare how energy efficient the storage is normalized by the mass, glycogen is very heavy. Lipids are lighter. So in a sense, you can pack up much more energy in lipids compared to glycogen. And considering that hummingbirds supposed to be constantly on the fly and they supposed to be moving very fast, it's much more efficient to like pack up a lot in a form of fat that you can use later than in the form of glycogen. So absolutely, they are doing it for the sake of like, let's store the energy to use it later. Like for example, overnight or for migration. But I guess hummingbirds have very specific preferences of the way of storing it in fat rather than glycogen.
Okay, thanks. So is there any other question? If not, I think we can wrap up. Just a reminder that the talk has been recorded. That this talk and all the other talks of the animal genomics meetings are available in the NFCore website. I share it with you in the chat. And yes, the other reminder is that we will stop now for the Christmas break and also for the pack that it's in January and many people it's going there. So that's why we are not having a session in January, and then we will start again in February. So as Francesca mentioned at the beginning, if any one of you are interested in in giving a talk here, or you know someone that is interested, please contact us here, I mean on Slack or by email. And yes, so thank you everyone for being here, and of course Katia for his very nice talk. Thank you everyone.
Thank you so much.
Thank you.